From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 30th, 2023. Hello, Micronauts, and welcome to today's Quality Quorum. I'm your host, Dr. Mark Martin of the Biology Department of the University of Puget Sound in Tacoma, Washington. Today is the 14th episode of Matters Microbial. Thanks for listening and/or watching. First, I'd like to share with you all a member of my Wunderkammer. What you're looking at here is a needle-felted tardigrade. It's no surprise I love tardigrades. They're an exemplar of living through challenges. And a few years ago, I commissioned this glow-in-the-dark needle-felted tardigrade from Marley and Myrtle at Epsi, because let's face it, everything is better when it glows in the dark. And here's the movie showing it doing so. I'll post a link to the artist's Etsy shop in the show notes. As far as today's inspirational lapel pins, at the time of this recording, tomorrow is Halloween, my favorite time of year, and in honor of it, a creepy jack-o'-lantern pin. Also, in a Halloween theme, one of my favorite quotes from George Orwell's 1984, "Reality is inside the skull." This still haunts me. I also wanted to share a wonderful pin that my wife 3D printed for me. Dr. Jennifer Quinn of the University of Washington-Tacoma, and my wife, is really a mathematician, but also a fine artist, and it depicts the Matters Microbial motto of mine across a streak plate. She's a wonderful artist. Next, thinking back to Dr. Ruth Eisenberg's wonderful presentation several episodes ago about the squid Vibrio symbiosis, one of my Micronauts a few years ago made this wonderful glowing painting of a squid and bioluminescent microbes. Creativity is important in all of my classes, and I continue to believe that it enhances learning. I do try to make my Micronauts appreciate how microbes are everywhere and do seemingly everything. So I assign them microbial pals and this sticker to give them some idea of the OMG, overwhelming microbial greatness, and WTM, what the microbe, all around us. Artwork by Ethan Kokek, and I'll include a link to his artwork in the show notes. As I've mentioned a couple of times on this podcast, having students learn about what lives in their reusable water bottles, their water bottle buddies, is a big deal. Here's a sticker that I try to get them to put on their water bottles. Artwork again by Ethan. Now, swabs of their water bottles plated on R2A medium, which is a medium designed for analysis of microbes in water, gives a variety of different types of microbes, as you can see. The next step is for the students to streak out one or two candidates for further analysis, and my Micronauts have gotten pretty good at it, as you can see with these restreaks. Then comes 16S ribosomal RNA analysis and very, very simple bioinformatics. Now, I adored in the past using the RDP classifier program out of Michigan State University, which gives wonderful confidence levels, as this image shows. But it's no longer available online, so I just have to resort to BLAST for now. Links to both approaches are in the show notes if you're interested. The goal, again, is to find out if close relatives of each water bottle buddy make sense that they be found there. For example, are they relatives of oligotrophs that don't use much in the way of nutrients? Are they biofilm formers that cling to the inside of the water bottle, etc.? And here are 6 quick examples from last year. You'll notice the real power of RDP classifier here because of the taxonomic confidence at each taxon. It does not mean anything scary has been isolated, however. These are simply relatives. Chrysiobacterium is a gram-negative bacterium that forms biofilms and is commonly found in soil and water. So it makes sense to find it in a water bottle. I see this organism quite commonly. Staphylococcus is a gram-positive bacterium commonly found on the skin and also forms biofilms. And since we handle our water bottles with our hands, perhaps that makes sense. Succomarella is a gram-positive bacterium found in soil and associated with humans. Not sure how that worked out for biofilms. But Sphingobium is a gram-negative bacteria found in the soil, and yes, in water, and often makes biofilms. Acinetobacter shows up from time to time as well, because it's usually found in soil and water. There are some relatives that can cause disease, but I don't think these do. And finally, and one of my personal favorites, Brevidomonas, a gram-negative bacterium often found in water, and it's a relative of Caulobacter, which is certainly an exemplar of living under oligotrophic conditions. Now, I have to tell you, and this is with deep, deep respect to our visitor today, that there are methods to do deep sampling and high throughput rather than just simply one isolate at a time, which is what we do. But I lack the resources, facilities, and funding for that particular approach at my institution. So the students learn techniques, a bit about identification, and it's a good place to start for them. And they feel really connected because everyone seems to have a water bottle for use. You know, years ago, I had a student in my class who seemed distracted. So I happened to say to this micronaut that we lived in a cloud of microbes. Why, I told her she was exhaling a personal cloud of microbes, as was I. What would happen when they meet? Clearly influenced by Charles Schultz's Pigpen character, she went right home and made me this piece of artwork. I didn't even ask her to. There are microbes literally everywhere. And that's a big deal to me. The famed evolutionary biologist J.B.S. Haldane was a tireless popularizer of science in the early 20th century. A newspaper reporter at the London Times supposedly asked him what, if anything, his research had told him about the mind of God. That God has an inordinate fondness for beetles, replied Haldane, famous for his wit. And yes, there are many, many, many species of beetles on our planet. But if God is fond of beetles, she must really adore microbes. And who better to tell us about how fundamental and ubiquitous the microbial world is around us than Dr. Jack Gilbert of the Department of Pediatrics at UC San Diego, as well as the amazingly beautiful Scripps Institution of Oceanography. Welcome to our Quality Quorum, Jack. It's such a pleasure to see you.
Thanks, Mark. It's nice to be here.
You know, it's so funny when I look at your background in particular, which covers quite a bit of ground. You've done so much, and that's why I think this JBS Haldane was perfect for you today. Would you like to, well, I mean, I can certainly read what you've done here, which is just amazing. You've founded the Earth Microbiome Project, the American Gut Project. It's just amazing. You wrote a wonderful book, which I'm going to put a link to in the show notes, called Dirt is Good with Rob Knight, that's been very influential to me. And so I try to think, rather than what does Jack Gilbert work on, what doesn't Jack Gilbert work on becomes the issue.
Well, I'd say, it's complicated, isn't it? Do I work on everything, or does it just look like? I'll say it probably just looks like that. I mean, it's interesting to note, obviously, and you know this, and most of the people listening will know this, that microbes are everywhere, right? So we can work on pretty much anything we want to work on if you put microbes into the equation. So as a microbiologist, we're all very well versed in our ability to apply the techniques and technologies that we learn and develop to the diverse areas of research which we're capable of exploring. I think from my perspective, I think it would be fair to clarify, right, that all of the work that we've done is deep collaborative effort, right? Often with extensive integration between different groups. So it's very hard to say that Jack Gilbert works on everything. I think I especially have been very cognizant of my own lack of knowledge and information in particular areas. And so it's always been a very deep connection with many brilliant collaborators. I'd say, if anything, any explorations which I've undertaken have really been at the behest of those deep collaborative efforts. The Earth Microbiome Project started when we gathered a group of 20, 30 people in a room in a mountain resort above Salt Lake City in Utah, right? And sat down and said, if you could explore the microbial world, if you could catalog it, how would we go about that, right? And I guess the two people in that room that really wanted to see that effort succeed in its immediacy were Rob Knight and Janet Jansen, with whom I co-founded that program. But realistically, there were another 18, 20 people in that room, all of whom are brilliant microbiologists in their own right. Ashley Shade's a great example, now out in France. And her research has really inspired a lot of the hypothesis-driven activities that we've done. So, I mean, saying that Jack Gilbert works on a lot of stuff is really just an indication that I know how to work with people. And I really, really enjoy working with people who are excited and engaged and willing to take risks, right? And those are the people I've searched out in my career and said, we could do this, it would be fun. And, the idea might sound crazy now, but maybe it could be extraordinary later.
Well, I mean, certainly you've been involved with all these different ideas, and every time that I've ever seen you at a meeting, you're constantly sparking off ideas and bringing people together. I certainly appreciate what you say, and it's important for many listeners and viewers to understand that a lot of science nowadays is truly much more collaborative than in the bad old days of Journal of Bacteriology in 1950 with one author. And bringing lots of minds together, especially with different points of view. And this is a real strength of yours, if you don't mind my saying. So may I call you a facilitator then?
Absolutely. Yeah, I
Yeah.
like that term, systems thinker, facilitator. When I speak to the team leads in my group and we're thinking about how different projects may connect, I can see things in my mind up above, and I make those connections, and I call it the mind map, right? So it sits there above my head, and I'm almost looking at it and thinking, okay, so if we do this, then this other piece of work can actually intersect, in 6 months' time once we've completed that piece, and all of these pieces come together, and that's, I guess, if I have a strength, it's the ability to see connections where other people might be not yet able to observe them. And I leverage that strength by surrounding myself with brilliant people who can actuate on those ideas.
Oh, it's absolutely true. And I mean, if you look at the history of molecular biology, the wonderful advances were made by people trained in physics. So in a similar way, I think bringing lots of different ideas, rather than a big tent, we can call it a big petri dish.
Sure.
But even that doesn't work since so many things are not cultivable. So it is absolutely true that we have all these things going on. So would you mind taking a moment? And I think the thing that most people would really, if they're not familiar with your work, would really bring people in is the American Gut Project. Just a couple of minutes on that.
Yes, the American Gut Project was started by Rob Knight back in 2011. And it was, he brought myself on and a number of other people on to create a citizen science project that would be paid for by people, right? So people donated their money to help create a database, a freely available database that had self-reported clinical information, health status, lifestyle, diet, etc. And embedded in that was the microbiome data, which we would generate with their generous donations. And so it's processed over 30,000 people. It's actually soon to relaunch under a different guise that would enable us to do this work. I do notice that balloons keep on going up behind me, and I'm not entirely sure why that is. But either way, maybe the balloons are good. It's a celebration, right?
Yes.
So I think that's really important, right? It was an effort to try and create an open source program of research that would facilitate us discovering novel insights about the microbes and the environments, and people's lifestyle traits, disease states, etc. And it's a fantastic resource. It's used by lots of organizations and research groups around the world to contextualize their own studies. And if you identify a biomarker that might be associated with a disease, you can go and easily download all of the American gut data and see if that biomarker is also associated with similar traits that are being reported by the American gut population. Of course, it also launched things like British gut and Australian gut, and and many other research programs around the world, which I think is the exact point. You create the infrastructure, you create the tools to facilitate people following the same kind of paradigm, and then everybody is empowered. The Earth Microbiome Project was exactly predicated around that idea. You know, Rob's a fantastic tool developer. There go the balloons again. Maybe it's every time I mention Rob. No, that didn't work. He's a fantastic tool developer, right? And he validates tools. He creates these platforms which other people can then use. You know, I would always say I'm a hypothesis-driven scientist. I like to think up ideas. I then like to test them. But we are more than happy to leverage the technology development that people like Rob have created that facilitate us being able to drive forward that research. That's not to mention all of the other brilliant people out there, you know, who have created platforms that facilitate our research. You know, Curtis Huttenhow is a really good example with his biobakery, and you know the many spin-off organizations and PIs that have left Curtis's lab. So there's those are the people that I really am indebted to. They're the people that create the platforms that facilitate us delivering great science.
Oh, I've certainly have met so many people that have worked with you, and I've been present at meetings where you've run discussions and all the rest. So I think American Gut's a great example of what the goal should be in open science, and that's good to see. I know that there have been people historically that maybe have had different points of view on this kind of subject, but I prefer the open science aspects myself. And I think most listeners and viewers would feel the same way because it's something that anyone can have access to and learn from.
Yeah, there are exceptions to that, though, Mark. I think it's important to recognise the need for data sovereignty for communities who may be underserved, who may have be worried that their data, their samples, could be exploited in a way that would provide them no benefit. And so I'm very cognizant of the need for improvements in data sovereignty, and the work of Keolu Fox, who's one of our professors here at UCSD, has really highlighted that for me, that we need to protect the sovereignty of data for communities, so that you know, from indigenous groups, from people who are underserved, to enable them to be able to access the intellectual property, the information, the data to support their community. So I think there's always a fine balance, right? If we're generating data from our communities in wealthy countries, in privileged groups of people, then it's reasonable to make all that data open access so it can help to advance science. But of course, there are situations where that's not necessarily appropriate.
I'm really glad to hear you say that. And I know from other things that you've written that you feel that way. I well remember the idea of looking for microbial diversity in certain countries overseas and then spiriting away what was found. And that always did bother me to some degree. But because of the sociological aspect, for lack of a better term, I'll just call it social justice because it is. I think that's a wonderful thing. There is nothing worse than taking advantage of those who can't. Punching down is a bad thing in science or anywhere else. So I'm delighted to hear that's what I already knew, that's a real emphasis of yours. So thank you.
Yeah, one thing we're
So
really keen about from the social justice perspective is also making sure that any new knowledge that we create in these environments can be used to address social justice and social inequity situations, especially if they have microbiome in mind. This is why I love the work of Professor Sue Ishak from University of Maine. She leads the microbiomes and social equity team. And that consortium is doing extraordinary things. In the journal I run, M Systems, for American Society for Microbiology, we have a special collection. I'll make sure that link is associated with this segment. But that special collection really does help to highlight the diversity of microbiome research that needs to be leveraged and taken into consideration if we are to make any real progress on improving equity more generally, but social equity and social inequity more effectively.
Well, I'll definitely include some links on that. And I had forgotten about Sue's work, which before I sat down here, I'm certainly aware of it. So I think it's important to put that up front, as you say. So thank you again for bringing that up. So you have yourself a group at an R01 PhD granting institution. You have all of these colleagues with whom you work. You work with them, not for they do not work for you. You work with them and they work with you, which is the way it should be. I thought it might be good to talk about a couple of examples of the kinds of projects that you're doing there. When I look over your wonderful website, and I will put a link to it, we have the human microbiome in nutrition, disease, and mental health. We have something that's near and dear to my heart, which is the microbial ecology of the built environment. And then we have marine microbial ecology in human and environmental health. And being associated with Scripps, that's kind of a no-brainer there, since I often think about what ends up in the ocean. Do you have a couple of projects within those three areas you'd like to talk about?
It's really hard to focus on one particular project because the people that lead them are extraordinary, and I really do view all of the research that we do as our children, if you will. So it's really hard to pick a favorite, right? I think the three key areas that we are super focused on, if I stick with human health to start with, is precision nutrition. So working with Gertrude Eklund-Mensah, who's a brilliant project scientist in our team, and Dr. Shokadera, who's a graduate student working towards his doctorate. We've been really exploring, and Joshua Tran, who's a new graduate student in our lab. We've been exploring ways to develop more effective mechanisms to predict which diets may be effective in helping to enable people to use food as medicine more effectively. So this is predicated on our $170 million consortium, for which I'm one of the co-chairs for the NIH, which is embedded in trying to develop a study population and to examine the impact of diet on that study population's physiology, with the understanding that the microbiome may be playing a role in mediating how people respond to food. And so with that study in mind, we've been building out and expanding in little ways and creating really exciting new research avenues. So the one I want to highlight in that is work going on with Gertrude Eklund-Mensah, where she is coordinating both dietary intervention studies. So seeing how people respond to consuming kombucha, which is a very, very Southern California kind of research project, and then also she works with Professor Lara Douglas from University of South Africa on a big international consortium that Lara developed called the Microbiomes Across Epidemiological Transition Study, the MET study. And so that program works with over 2,500 people of African descent, trying to understand how diet influences their physiological and cardiometabolic disease state and burden over decades of observation. And then we got a really fun one that we're doing. If you ever had a colonoscopy, you'll love this one. So we Joshua Tran, who's a graduate student in our lab, studying for his doctorate, is running a new clinical study, and will be recruiting soon, for people undergoing their second or third colonoscopy, and we're going to give them a green Mediterranean diet, randomize them to a green Mediterranean diet after their colonoscopy, to see if we can remove certain bacteria that we've identified that can be it can exacerbate the risk of the onset of colon cancer and polyp formation, precancerous polyp formation in the colon. So we're trying to investigate ways of using food as medicine in that respect. The other human one I love is our gut-brain axis research with a new graduate student. Well, not new; she's been here 2 years. Called Dottie or Marisol Dothard, and Marisol's doing fantastic work trying to understand how probiotics could be used to treat depression and anxiety in animal models. And she's also exploring this in the context of trans men, so women who have transitioned and are undergoing testosterone therapy, and the implications for hormonal disease associated side effects. So onset of vaginal disorders associated with testosterone utilization to initiate transition, and then effective strategies to understand how to mitigate neurological side effects associated with those transitionary phases. So there's a whole suite of little great research programs that are ongoing in that area. Oh man, I've got so many ideas. It's always really hard to contain them.
So it's an interesting idea because, and again, that's why it was a challenge for my wife Jenny and I to come up with a good title for you for that reason. And I know that's presumptuous to give you a title about this, but I did think it was one of the things about you is you have such broad interests, and you and I have talked about that before. And there's a big push not to have broad interests in some sectors of science. And what I hear from you is the fun of what you do.
Yeah. I
And
love what we do because it's fun. If it wasn't
right.
fun, I'd stop doing it. And I think maybe because I have an ability to continuously focus on multiple things at this in very quick succession. I never say at the same time, but quickly switch between ideas. And I get bored if I don't do that. So I really do like the idea of having 50 ongoing research projects and constantly switching my attention to them. One of our fun ones in the built environment now is we're starting to print building materials that contain bacteria in them so that those bacteria can kill off pathogens that may land on a surface, say in a hospital. But also, and these are fun, so that the bacteria in there can stimulate immune responses to dampen down systemic inflammation through the skin or through respiratory breathing. And that strategy, I think, is one that we're really keen to explore, how to make buildings healthier for the occupants. We've actually thought long and hard about the potential implications of applying probiotics, if you will, into building materials. The organisms that we have started exploring this phenomenon or this mechanism with are generally found in built environments anyway. What we're doing is we're providing a substrate which facilitates their growth and activity against other organisms which we know to be potentially harmful to the occupants of that space. We are being extremely careful in trying to understand the implications of applying these organisms. So, for example, it's possible that some of these probiotics that we put in the building materials could acquire antibiotic resistance from known pathogens. It's possible that they could have unknown interactions which could lead to negative health outcomes. So we are doing a lot of validation and baseline work because we're not a company, right? We're not trying to get a product out there. We're taking our time and doing the ethically responsible research to be able to understand what the true implications of this kind of technology could be. The flip side of that is we found that some of these bacteria are really good at binding human pathogens and human viruses. So, for example, some of the organisms that we're deploying can actually bind SARS-CoV-2 and prevent it from getting inside the body. And so we're exploring these phenomena and trying to see if they can be viable mechanisms to help reduce the epidemiological spread of disease in buildings and environments that we interact with every day. And another one, just for fun, we're doing a huge amount of work, obviously, with oysters. I don't want to say obviously. Why would it be obvious? But in the context of this, I have a whole research team, Emily Kunstelman, Cara Wigan, and researchers from across the board in their group that are focused explicitly on trying to understand how climate change will impact oyster industry with the enhanced viral predation of oysters, and also how it could influence human health and epidemiology from foodborne transmission. And so those Emily and Cara are exploring this, and then taking it in December to the UN, to COP28, the climate program in the UAE, or Dubai, I can't remember where it is now, but to explore how to gain research interests from the rest of the community and apply those into policy, so that we can not only reduce the potential impact of climate change upon epidemiological disease transmission in foodborne pathogens, but also protect a burgeoning high protein industry in the US and abroad, by being able to predict and mitigate the effect of viral disease on oyster stocks in fish farms. So we're really, those kinds of project ideas, they come from sitting down with people and just chatting about what their interests are, and then working to help facilitate those by connecting people to each other in a way that facilitates exciting research.
In something, and I know that you're you don't particularly like being praised, but I think this merits stating, is that the number of times that you mention the people who were working with you as your equals, it's really quite remarkable, and it needs to be noted, because not everybody does that, and I understand a certain degree of that. But this is something that you've always been about as long as I've known you, is being collaborative and making sure everybody gets credit for what they're doing. This factors into your feelings about the impacts of your research. It's just part of what it is to be Jack.
I mean, I'm a very privileged person, and I'm fully aware of my privilege. It is not hard to be a white male with an English accent in America. It really isn't. And while especially as the older I've got, I've become aware of what my skills are and how I can apply them, and I try and do that in a responsible and equitable fashion. I also know what my weaknesses are, and so for me, working with people who can bring their skills, their amazing talents to the table, and we can work together to do something even more extraordinary than we'd be able to do alone, is great. And to a certain extent, I don't need recognition. I mean, I do like recognition. I'm not a totally selfless human being. I just won an award, and I was like, woohoo, I won an award. That's fantastic. I really liked winning an award. It's nice to be recognize every now and again. You know that, everybody knows that. It feels like you might be doing something, you might be going in the right direction, if you know what I mean. Sometimes
Yes.
without that kind of feedback, it's hard to know. But to be perfectly honest, the best thing in my life is seeing other people succeeding and having and doing exciting work and amazing research. One of my graduate students, Sean Gibbons, is a great example of that. He's up at the Institute for Systems Biology in University of Washington. He's done extraordinary career progression development up there over a very short period of time, and is really becoming a global leader. And honestly, that makes you, it warms your heart, right? Like to say that
Yeah.
I had any association with that person and could help in any way to facilitate their development. That's really, it's like kids, right? You want your kids to do well. And so for me, that's the biggest reward, is seeing people do well, seeing people happy, going out of my way. I mean, I meet with all my students on a weekly basis, and I'm always available to them. To me, their success and their development and their support is the most important thing. And that's the reason, I do what I do, is because I want them to be successful. I want them to get out there in the world and make a change, and do things that will push back the frontiers, especially for equity in our society.
Sean, is going to be on the podcast in about a week, I think. So he'll be
Great.
a nice follow up to you, actually. He's always been quite wonderful to me. So there you go. I think you have created other people and encouraged them to have some of these characteristics that I think are good for all of science. And I think that's true.
I mean, people, I say life is too short to worry about all of the difficult things in life too often, right? Obviously we all have difficulties and we deal with those as we see fit, but especially in science, there's so many exciting things to do. I just want, if I can help to create an idea and then watch other people take it, that is absolutely perfectly fine by me. There's way too much stuff to do to sit down and feel like you own something. And I've never
Well,
really got that in people. There's a lot of people that feel like, well, that's my area of research, and everybody should, I know nobody should be competing with me. I hate competition. I know some people think competition drives things. For me, competition it can be ugly. I much prefer just doing fun things and working with people and seeing what we can do together.
I have a saying about the microbial world, first evolved, last extinct. And one thing about that is that whatever it is that you might have uncovered in the lab was already there.
Mm.
You didn't make it happen.
Right.
Now, I don't know, as synthetic biology, improves, maybe that won't always be that way, but that's certainly how I've felt. And I get just as excited by silly stuff I see in my little lab as anybody else does. And that's really what it's about for me. And I just want to underscore what you said. Watching people with whom you've worked do wonderful things is the best reward. Because so many times it's like living on Yelp, where people are perfectly okay saying every negative thing, but very, very rarely do they want to say positive stuff. So I view every successful student that I've had as just about the best reward I could get.
Yeah, and the leaders in my life that I've most looked up to have followed that exact mold, right? I'm here to support the next generation and to do fun
Mhm.
things along the way, right? And I think especially with global climate threats and with social inequity in our society, anything we can do to try and create resiliency both within our society and within our infrastructure, and create positive change to facilitate operations that can build that resiliency, is going to become not just valuable but essential to the survival of our species. So I feel like it's axiomatic that we really do need to sit down and work on this, right? And work together, and any negativity should be excluded from that equation. But
Well, I mean, we're all in this together, and the idea of helping others is something that we all say we really believe in, but we have to work at it because
right,
we've got our bad sides. All of us do.
absolutely,
But
yeah,
but the positive results that I've had from reaching out to people and helping them, or at least being present when they've done well, very hesitant to say that. I've got 28 of my research students who've gone on to get PhDs. That doesn't mean I made them do it. It doesn't mean that I'm responsible. What I am is a talent scout,
yeah,
and that's okay. I like being a talent scout. I'm pretty good at it.
yeah.
So it sounds to me like you've taken it just exactly the same set of ideas and done really good things with it. And it's just been a marvel to hear.
Oh, yeah, thank you.
So do you mind if I ask just a couple of questions and then we can think about closing out?
Go for it.
So the first is there anything you've said so many things here. I was going to ask you maybe for a list of links about your ideas about, social equity and microbiology in particular, because I can go looking for them and I will, but I know this is something near and dear to you, and I think it would be a good service to the listeners and viewers.
Yeah, we'll make sure you have those, and they'll be linked in this cast.
That's really, really good because I think that's important to know. And because again, these are things that we talk about often, but we really need to make an effort to think about them. And you've certainly been very useful to me today in this subject. Secondly, what is the one thing that sold you on being a microbiologist, Jack?
I like to think about this all the time. The one thing that did sell me on being a microbiologist, which is really weird because it took a while to kick in, was it was the first textbook that arrived when I finally got my university position as an undergraduate. So I did really badly in school, not utterly terrible, but I nearly failed the exams that would allow me in England to go to university. But I was given a chance by a university, King's College, and they took a risk. They let me in with poor grades, and I got that one textbook through because we had to get our books before we started school, and it was the first one to arrive. So I thought, I'm going to do my best here to try and pay back the support that the university had given me to become to take advantage of their generosity. And so I read that textbook, and I don't think I'd ever read a textbook before because I wasn't the most studious person. And it was eye opening. I don't think I'd ever really thought about microbes before that, and I was 18 years old. And so I sat down and I read that textbook, and it changed the way I think about things. I didn't really follow up on microbiology too much for the rest of my degree, but when an opportunity came after my degree to do microbiology, I thought, yeah, I remember that. That's super exciting, and there's some really cool stuff going on. And so I think textbooks, while we pour derision upon them to a certain extent, are one of the most valuable ways you can reach young minds, right? If you can create a textbook that's impactful and has really cool stories in it and really cool graphics, etc., it can change the world in interesting ways.
Oh, that's absolutely true. I always like to ask guests that because I'm really curious what got them going. I've also noticed that you've been all over the place in your career. What is the most unusual place you've done microbiology?
Months, so surrounded by a bunch of service personnel who become like brothers to me. But at the time, it was very isolating and an extraordinary opportunity and a privilege to be out there in a very isolated part of the world, doing microbiology, which was applied. Right, applied microbiology has been incredibly important to me. I'm currently the president of Applied Microbiology International, which is the oldest society for microbiology in the United Kingdom. And doing that work really opened my imagination and understanding to use-inspired science, to say that I can do my science, I can do basic research. And the research I did there, 15, 20 years later, under the auspices of many different graduate students and postdocs over those 2 decades, came up with some really interesting findings, which I'd need a little while to explain. But at the time, it was all use-inspired. I was working for a company. And so it was a really interesting part of my life, learning how to apply microbiology in a way that would actually change the world and have an impact upon everybody.
That is an amazing story. Normally when people make fun of microbiologists, the exciting thing that they do is put a cover slip on a slide or something similar to that. I would hear this from ecologists all the time, but it's not true. A lot of microbiologists that I know have been fortunate enough, privileged enough to visit some really unique places. Even for me, and I'm not very well traveled, when I first went to Delft, and could stand where Leeuwenhoek stood. I mean, that meant so much to me, right?
Yeah.
So it's the really wonderful thing about being a microbiologist. We're all not like bass specking, that everything's everywhere, but to some degree it is.
Yeah, I love
Jack.
Hawking's hypothesis. So I've spent
Oh
my
yeah.
life trying to prove that in certain situations. But the ability for microbes to distribute around the planet is phenomenal, right? And localized evolution is a real thing. Absolutely, selection is a real thing. It was part of the hypothesis. But I honestly do believe, in the context of global ecology, that bass specking was probably right.
I'm certain that we're going to find spores or something similar up in those Lagrangian points too.
Yeah.
But we won't need to talk about that. I've gotten people angry with me with thoughts about that, but we won't need to go into that. But it's always a pleasure talking with you, Jack. I feel energized every time that we do, and I hope that our viewers and listeners do the same thing. Please give your coworkers and your family my very best, and thank you again, Jack.
Absolutely. Thanks, Mark. I appreciate it.
You're very welcome. And now I'm going to do This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. show notes, and as always, with remarkable links to visit, for today's episode can be found at microbe.tvmm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Jack Gilbert can be found in the Pediatrics department at the UC San Diego, as well as the Scripps Institution of Oceanography, which I miss. Many, many thanks to David Renata for superb editing and Rupert Clark for this wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next week on Matters Microbial.